few of you out there will know me, Mark Worrell. I'm one of the accreditation team leaders at NATA. Less of you might know that I'm trained WADA international sport laboratory assessor as well. So I go around to laboratories and assess the WADA laboratories that we have. We have 4 in total under NATA's watch. So a few of you here today would be forgiven for thinking that Accreditation Matters 24 is the only event happening this year, but it's not. Currently over in Germany, England are poised to win the European Football Championships, and in 31 days' time, the Summer Olympics start in Paris. Our next 2 speakers are experts in keeping sport fair and protecting the competitors, be they human, equine or canine. Our first speaker is Dr. Judy Edmonds, the Quality Manager of the Australian Sports Drug Testing Laboratory, or ASDTL, just up the road here in North Ryde. They're part of our National Measurement Institute. Judy has a busy job of keeping both NATA and WADA happy. And Judy's talk is entitled Testing Times, the Interplay of WADA, NATA and ILAC in Accreditation. Following Judy will be Dr. Adam Corley, the Scientific Manager at Racing Analytical Services in Melbourne. Adam started his career at the ASDTL and was heavily involved in testing at the 2000 Sydney Olympics. Adam has since added animal sports to his expertise and is also a NATA technical assessor. Adam's presentation is entitled Winners and Sinners, Accreditation Matters in Forensic Drug Analysis. To kick off the session, however, we have a brief message from someone who will feel very much at home during the Paris Olympics, Professor Olivia Rabin, the Senior Director, Science and Medicine at WADA.
The MOU between ILAC and WADA is important. It's because the anti-doping laboratories that are accredited by WADA are operating under a double accreditation, the ISO 17025 accreditation and the WADA accreditation. So it is very important that the accreditation bodies speak to each other so that we can harmonize the rules and harmonize the way we audit the laboratories to ensure that there is a good communication between the 2 standards. So it's very important to have this MOU in place so that ILAC and WADA can openly discuss and harmonize their practice when they audit anti-doping laboratories. The athletes around the world request from WADA that their samples can be tested with equal quality all around the world. It's important for them to know that wherever their sample is going to be analyzed in any WADA accredited laboratory, the same quality of analysis can be applied. So it's very important that we can operate with a high quality, and of course the way we establish our standards, the way we apply our standards, the way we audit the laboratories, both from the national accreditation bodies and WADA, is extremely important in the way we can harmonize practice between all the anti-doping laboratories around the world. The Australian anti-doping laboratory is part of the 31 anti-doping laboratories accredited by WADA around the world, and of course the same rules apply all around the world, including in Australia. Well, the Olympic and Paralympic Games are very important competitions around the world. They are very visible, and we know that the laboratories, in particular during the Olympic Games, are put under a lot of pressure to analyze a lot of samples in a very short period of time. So this is important. Having said that, accreditation is applying all year round to all the events, sport events around the world, should they be Olympic Games, Paralympic Games, Continental Games, World Championships. So it's very important for WADA that this quality can be maintained all year round, regardless of the level of the competition. But certainly the Olympic Games is an important moment, not only for the anti-doping labs, but for all of us who are sport fans.
Hello, and thank you for NATA for inviting me to speak today. All right, so the Australian Sports Drugs Testing Laboratory holds dual accreditation, both for the WADA ISL which is the international standard for laboratories, and for the ISO 17025 testing and calibration standard. We hold a flexible scope of accreditation, which allows WADA labs to update and test for new substances that have been added to the WADA Prohibited list. That gets updated every year, so we need to be able to keep on top of our testing. We also can upgrade our instrumentation on a regular basis so that we keep at the forefront of being able to analyse samples and be able to test for the prohibited substances. WADA labs analyse both urine and blood samples for substances and methods that are listed on the WADA Prohibited list, and this is done in accordance with WADA technical documents and technical letters, which I'll go into detail in a little bit later. So for labs to actually gain WADA accreditation, firstly, for a candidate laboratory, they must provide an expression of interest to WADA, and the lab must have support from a host country that has an anti-doping program, and then support from an institution such as a university or hospital or a government organisation that is separate from the anti-doping organisation to be able to do the work. They must ratify the UNESCO Convention Against Doping in Sport, provide a detailed business plan, and have compliance with the WADA Code of Ethics, which in part is also to do with confidentiality and impartiality. The candidate labs then undergo a pre-probationary test, which is analysing samples that are provided by the WADA Proficiency Testing Laboratory, and then they also undergo an on-site WADA assessment prior to becoming a probationary lab. So if they pass the pre-probationary tests, those candidate labs become a probationary lab. The probationary labs then participate in the WADA External Quality Assurance Program, which is known as EQUAS, which all WADA labs are part of every year. They must plan and implement research and development into anti-doping science and be willing to share that knowledge with other laboratories and anti-doping scientists. If they don't already have it, they must obtain the ISO 17025 accreditation, provide WADA all of their validated methods for mandatory tests that are stipulated by WADA, perform a final accreditation test, which is testing 15 EQUAS samples and report the results within 7 days, so to mimic doing a fast turnaround time assessment. And WADA may then recommend that lab be accredited, and they'll then issue an accreditation certificate. So once a lab has gained accreditation, they must also maintain their ISO 17025 accreditation, participate in the WADA EQUAS program, which is 3 rounds of 5 samples each, where we know they're coming from WADA, and then there's a further 5 samples that actually come through our anti-doping partners to mimic samples being collected from athletes. The labs must maintain their independence, impartiality from the anti-doping organisations, comply with the WADA Code of Ethics, and continue their research and development activities. They must maintain testing of a minimum of 3,000 samples per year, and also then be assessed by an accrediting body such as NATA, and WADA also come and do on-site assessments. So on the WADA website, there are actually 30 labs that are currently accredited for doing both urine and blood analysis. There are also 2 further labs that just do blood analysis, and this is because analysis of whole blood samples must be done within 24 hours of collection of the blood, and so the samples must be sent to the closest laboratory. So there are 2 extra labs to make sure that the testing can be done within that time frame. So our anti-doping partners, along with WADA and NATA, we collaborate quite closely with other WADA laboratories. We also rely on our NMI laboratories who provide us with certified reference materials, both as pure substances and in matrix, and that's quite important in our testing. We work with anti-doping organisations such as Sport Integrity Australia, Drugs Free Sport New Zealand, and the Oceania Regional Anti-Doping Organisation. We work with the sample collection authorities who are responsible for actually going and collecting the samples from the athlete, and then maintain the sample collection authorities need to maintain a chain of custody of that sample then being delivered to the laboratory. We work with testing authorities who are responsible for actually planning and implementing the testing of athletes, and that is in conjunction with the results management authority, they're often the same organisation, who then, once they've received the test results, determine whether further testing is needed or the notification of adverse analytical results. So the testing and results management authorities can be organisations such as Sports Integrity Australia, but also international federations such as World Aquatics or FIFA are also responsible for those. So ASDTL can only accept samples for anti-doping testing from these organisations. We can't accept them from athletes directly or the general public because that contravenes the anti-doping testing. So all of this is managed by the Anti-Doping Administration and Management System, which is known as ADAMS. So each of the modules are separate. So we as the laboratory representatives, we can't access the athletes module. But what the athletes can do is they can see their sample numbers for the samples that have been provided to laboratories. They can also update their whereabouts of where they're going to be at any given time. So if they've got to change their whereabouts because they're going to compete shortly, they can update that. That information is really important for the testing authority to then plan and implement the testing of those athletes. And that's done in conjunction with the the results management authorities who are then deciding whether further testing is required of those athletes. The laboratories upload into our portal the results of any analysis which the TAs and RMAs can access. And the final module is the Athlete's Passport Management Unit, which is part of a select number of laboratories, and that looks at longitudinal results of athlete testing, which I'll go into in a bit more detail. So the WADA prohibited list is divided into classes of prohibited substances. The majority of them are prohibited at all times, which is also known as an out-of-competition test. A further subset are substances which are only prohibited in competition, and the in-competition period starts at 11:59 p.m. the day before the competition, up until the end of competition and any subsequent collection of samples directly after that competition. There's a further subset of samples that are only prohibited in specific sports. These often are to do with sports like archery and shooting, where the substances in this class are also can be in competition or out of competition prohibited, depending on the sport that they're in. So there's a lot of accreditation and that needs to be managed. Technical documents are issued to provide specific technical and procedural requirements for testing of certain analytes or methods. They are mandatory documents, we must implement them, by the effective date that is listed for each document. They do get updated periodically, new ones get added, or occasionally some of them do get removed. In association with this, we also have technical letters which often provide further guidance, particularly on certain analytes that we test for. Again, these are mandatory documents that we have to work with, and they often, if it's an updated one, they are implemented immediately, and if it's a new technical letter, there's usually an implementation date that we must meet. So just a brief sample analysis. So the sample comes to us with an A and a B bottle. It's all de-identified. We don't know who the athletes are. They arrive in a tamper-proof container. We only analyse the A bottle, so it comes as an A and a B. We open the A bottle. Most of our techniques use either liquid or gas chromatography coupled with mass spectrometry. And as an initial testing procedure, we test for over 400 different analytes within that sample. If no prohibited substances are found in that initial testing, the sample is reported into ADAMS as a negative. If we find or suspect a prohibited substance is present, that's sample, another aliquot is taken, and that is sent to confirmation procedure, which only tests for that specific analyte and any associated ones according to any technical documents or letters. So the outcome of the confirmation test is that we can either report it as no prohibited substances found. If a prohibited substance is found, we report it as an adverse analytical finding, which would often be reported in the media as a positive. The other option is also for an atypical finding, and this often is where, for example, there are substances that are produced naturally in our body that also can be used in a performance-enhancing way. So sometimes there might be atypical results that result from that. So they're the three main types. And normally with an atypical finding, the testing authorities and result management authorities will, or the lab will advise to do follow-up analysis, or they can also request for further analysis to be done on the sample that we already have. A little bit about the Athlete's Biological Passport, which is a longitudinal testing program that has a haematological and sterile profile module. The data from every single sample, either a whole blood or a urine, is uploaded into the respective module of the ABP, and it's allocated to the passport for that athlete. So every sample is then tracked longitudinally for each athlete. The passports are then managed by the APMU, and APMU is a separate WADA accreditation, which ASDTL does hold. Not every lab is an accredited APMU. And so the analysts within our laboratory who are members of the APMU then review passports for signs of atypical results. So it's a way of just checking that there's no underlying doping going on. So accreditation matters in anti-doping because an elite athlete can be tested numerous times throughout their career, and those samples from that athlete can be tested in various laboratories around the world. So there needs to be a consistency and harmonisation across all of the laboratories, not only in the analytes we test for, the way we test them, and also the instruments that we use and how we interpret the results. And it gives confidence that anti-doping testing is working, and that gives confidence to athletes and the general public that the results of elite athlete sport is not influenced by performance-enhancing drugs. We also need to have traceability of our results because the results of The analytical testing can be used in anti-doping rule violations that are presented to the Court of Arbitration of Sport. So it is a legal requirement as well to our testing. So we need to maintain quality assurance in our staff training and expertise, our adherence to documentation, our risk management, and always be looking for ways of improving our lab practices, our methods, and our staff training. So coming up to the Paris Olympics, we are currently being provided with samples from our testing partners from athletes who have been selected or potentially selected for Australian and New Zealand Olympic teams. And we have to analyse those before the athletes are allowed to go to Paris. We also have been testing samples that are currently held in long-term storage. So long-term storage is a way of deterrence. So we can test samples retrospectively to see if they comply with anti-doping rules. And so we've been completing that in the lead up to the Games as well. And we also have 2 analysts who are going to the Olympic Games and 1 analyst going to the Paralympic Games to help with the testing and analysis of samples to help meet the turnaround times that are required for an Olympic Games. And finally, I'd just like to say thank you to everyone for listening and also to the members of ASDTL. Thank you.
Thanks, Judy. Judy's given you a great overview there of human anti-doping, and I'd like to now give you a little bit of a background in what happens in animal anti-doping. So, winners and sinners. I'm the Scientific Manager of Racing Analytical Services Limited. I'll just begin by giving you a little bit of a background into our rather unique company here in Australia. Established in 1988 by the Victorian State Government and Victorian Racing Industry, or VRI. We've been accredited by NATA since 1992. And in that time, the company's grown from a staff of 8 to now we've got 61 employees. We're an independent, not-for-profit company limited by guarantee and quite a unique structure, which has attracted me to the great weather of Melbourne. Why I've moved down is that we've got a RASL board made up of 4 independent directors appointed by the Victorian Racing Minister of the day, but also 1 director nominated from each of the VRI codes. That's Racing Victoria, Thoroughbred Racing, Harness Racing Victoria and Greyhound Racing Victoria. Now, our services obviously are primarily for doping control for racing authorities. We're now the largest racing laboratory in the world, out of about 50 laboratories around the world, and are receiving approximately 57,000 racing samples per year. That's comprised of all 3 racing codes, obviously, in Victoria, which is about 50% of those samples. Also, all 3 codes in South Australia and Tasmania. Greyhounds from here in New South Wales and Northern Territory, and thoroughbreds in Chile. The company's also always had a strong presence in non-racing work in order to derive external revenue and keep the cost of service for our racing clients, particularly the VRI, at a minimum. And now we receive approximately 20,000 samples a year not related to racing. These are human workplace testing, 4308, and oral fluid 4760. Human hair testing for sporting and medico-legal applications is a rapidly growing part of our business over the last 8 years. And also testing and certification program for sports supplements under our HASTA program. That's Human and Sports Supplements Testing Australia. So why do we test for drugs in racing animals? Well, really there's 3 equally important aims. That is welfare and safety of animals and their riders or handlers, the integrity of racing and the fairness of competition, and also the protection of the breed. Now, to put this in perspective, the racing industry, at least just in Victoria, and you can extrapolate nationwide and then internationally, the contributions and responsibilities of the racing industries in Victoria, estimated approximately $4.7 billion to the Victorian economy, more than $500 million in taxation revenue. The VRI employs an estimated 120,000 people, or 35,000 full-time equivalent, and greater than 50% of the economic return from racing is in regional areas. However, as you've noted, no doubt, in recent years, we operate with a social licence to ensure integrity and animal welfare, and hence those 3 aims that I just described. How do we do it? I don't have time today to go into a lot of detail. I will point you, even though it's 10 years old, to a very good article written by our colleagues in Hong Kong, Jenny Wong and Terry Wan, for a guide for clinicians in the Veterinary Journal, but covering everything from prohibited substance categories all the way through to reporting and international harmonisation. So good bedtime reading for you all, an 8-pager, but quite a nice article I've often referred to. Of course, we're here to talk about laboratory accreditation, and we're accredited by NATA, assessed for compliance to ISO IEC 17025, the general requirements for the competence of testing and calibration laboratories. Interestingly, racing laboratories around the world, and as I say, there's approximately 50, ISO IEC 17025 is interpreted by an ILAC document, a G7, and that was quite unique. There's one now for WADA accredited laboratories, but this has been in place for some years now, and in fact Mark is involved with the writing and review of this particular document. As Judy alluded to before, it's so important that we operate under a flexible scope of accreditation to remain up with doping threats in both human and animal sports. And under the ILAC G7 document, that's what provides us with that coverage for flexible scopes of accreditation. What this is all to do is to support the International Federation of Horse Racing Authorities, or IFHA, international agreement on breeding, racing and wagering. So this is a harmonised set of rules across the world for thoroughbred racing. And really what it comes down to us in the lab is to be able to detect substances capable at any time of causing an action or effect in the mammalian system. So it's a very broad remit that we have, basically to detect the presence of any pharmaceutical in the animal's body. Also, what we have at RASL is IFHA accreditation, another layer. We obtained this in 2015 and now a part of one of just 6 laboratories in the world, with LCH in France, LGC in the UK, UC Davis in the USA, Laboratory of Racing Chemistry in Japan, and the Hong Kong Jockey Club Racing Laboratory. What the aim of this is for the IFHA is that, in theory, any Group 1 race, such as the Melbourne Cup, the samples coming from such a race would be analysed by an IFHA accredited laboratory. What this has been made possible and supported by is virtuous funding, and as I say, our company model really assists that. Also dedicated R&D program where we have 6 full-time equivalent staff in R&D at RASL. And there is also, as you would expect, additional proficiency testing requirements. I thought today was an interesting opportunity to let you know that times have changed. In my opinion, just 10 years ago, customers, that is stewards of racing, for example, and also participants, being racehorse trainers, where strict liability rests, through their lawyers, did not show a great interest in NATA or laboratory accreditation. You might find that surprising. We, as analysts in inquiries and appeals, would want to talk about our accreditation with NATA and didn't really get much of a response. But since high-profile cases involving cobalt findings, particularly in Victoria over the last 10 years, there is now a greater awareness of NATA and the importance of accreditation, supporting confidence in the results provided by racing laboratories. And common questions that I might field, in fact, just last Wednesday in the Victoria Racing Tribunal, the case, Dr. Corley, is RASL a NATA accredited laboratory? Can you please explain the process of accreditation? And was the sample in question analysed with an accredited method? These are quite common questions now, and that we need to be able to explain them to a lay audience. Racing operates in an interesting paradigm. Judy's gone through in the human anti-doping sphere. It's all about performance enhancement. In racing, we also need to, again, detect the presence of any pharmaceutical in the mammalian body. That can also come to performance impairment, which might result in betting manipulation, and the interplay with legitimate therapeutics, the so-called medicine box, that can and should be used to look after the health of competing animal athletes. Of course, what this provides is a very large chemical space that we need to analyse, ranging from anabolic agents, which might be neutral in their chemistry, right through to trace elements analysis. Just to give you a little bit of an overview of our sample workflow, human and animal anti-doping laboratories over the last 25 years have been very effective in automated solid phase extraction techniques and being able to then separate subclasses of compounds into various screens. In animal testing, or for horses at least, we also have total carbon dioxide in equine plasma by headspace gas chromatography mass spectrometry, cobalt and arsenic by inductively coupled plasma mass spectrometry. Since the peptide scandals in football codes here in Australia 10 years ago and also around the world, peptides has been a growing problem for us to analyse, and more recently animal hair testing laboratory or animal racing laboratories have invested considerable effort in hair testing. Also, we still conduct erythropoietin analysis by ELISA screening and LCMS confirmation. Within this framework, so the vast majority of that testing is qualitative in nature, the presence detected or not detected in a sample, and I'll speak about the criteria for that in a moment, but there are a number of quantitative requirements, threshold substances for substances that are endogenous, naturally occurring in the body, ranging from total carbon dioxide in equine plasma to control the misuse of alkalising agents, right through to ethanol in canine urine for that reason of performance impairment I was saying earlier. Of course, each of those then need to be validated and also with an associated estimate of measurement uncertainty for accreditation. Proficiency testing is very important, and this is conducted under the auspices of our Association of Official Racing Chemists, or AORC, an annual single blind assessment with urine, plasma and hair substances, but also a quarterly inter-laboratory exchange of negative samples. And this is quite a neat program that our customers, being the stewards of racing, and participants like to hear that in fact cleared samples are also tested by another accredited laboratory. The AORC guidelines are very important for, I mentioned earlier, qualitative criteria, what allows us to in fact confirm unambiguously the presence of a prohibited substance in a sample. With reference to an authentic reference material that often may come from the NMI here in Sydney. Concurrent criteria of minimum of 3 diagnostic ions or transitions from multiple reaction monitoring, signal noise ratio greater than 3, and with the advent of high resolution mass spectrometry, mass accuracy of within 5 ppm or 2 millidaltons. And again, I don't have a lot of time today, but just to give you a background, LC-HRMS, high-resolution mass spectrometry, has really been a game changer for us in anti-doping over the last 20 years. When I think back to 25 years ago, what our aims were, at least then when I was in human anti-doping, and what now we're able to achieve with the use of high-resolution mass spectrometry, it's really quite impressive. And for those of you interested, a nice review then there from my good friend Alexander Makarov, the inventor of the Orbitrap mass spectrometer. And what this enables to do is to address the capability gap, or at least try and minimise it, between the number of drugs available, which increases every day, to those detectable. And as I say, for racing laboratories, that's a big job. One of the areas we've invested in, as I mentioned earlier, is animal hair analysis. We've been accredited by NATA since 2017 for that. Now, the most significant potential benefit, I need to say, however, is that the potential for extended detection period of permanently banned drugs that hair can offer. And it's one of the important nuances it's important to relate to a lay audience or an informed audience such as this, is that hair really needs to be considered as a complementary matrix to plasma from blood and to urine. They each have their place, as I've provided here in an encyclopedia chapter. There's been a common misconception, in my opinion, for many years in human and animal anti-doping that blood is better than urine, and now hair is some sort of silver bullet, and that's just not true. Each matrix has its strengths and weaknesses. What that experience allowed us to do as a company was then expand into human hair analysis. Unlike the other urine and oral fluid, there's no Australian standard for hair testing yet, but we operate under the Society for Hair Testing, or SOT, international guidelines for harmonisation. And HASTA has 4 separate methods covering drugs of abuse, cannabinoids, anabolic agents and ethyl glycuronide. And as I say, this is applied for medico-legal and applications. What our R&D in small molecule analysis at least has enabled is we were the first laboratory accredited by NATA in Australia for investigative testing of miscellaneous samples. So these can range from anything from filled or empty syringes, tablets, vegetation, pills, capsules, any number of paraphernalia that might be seized from the property of licensed persons in racing. That could be, of course, kennels or stables, but it can also be their car glove box, literally their kitchen sink. And what we, as well as analyse in a targeted regime these samples, we also perform untargeted screening using sophisticated MS libraries, so semi-targeted screening, but also in silico technologies we're working on quite heavily. Now, while the laboratory approach is undefined, considerable guidance is given in this flowchart design developed by Dr. John Vine, with our SOP of 22 pages covering everything from sample receipt and chain of custody, which obviously is very important from a legal sense, right through to the results interpretation and reporting to customers and any follow-up work from such samples. Also, what that work enabled us to do as a company was expand into supplement testing, where some athletes use supplements, of course, to maintain dietary needs. Manufacturers, though, might use novel ingredients for market advantage in this multi-billion-dollar industry, and so for athletes to have confidence in supplements that they may use, offers a certification product under our HASTA brand. They're looking for more than 250 substances included in the WADA Prohibited list that Judy spoke about earlier. And then consumers can cross-check tested batch numbers with our website and the Sports Integrity Australia app. Now, what's important, of course, with accreditation is also people and culture. NATA, of course, will focus on training records of our staff, but I can tell you that it takes 1 to 2 years on the job training for our certificate or diploma qualified technicians and degree or PhD qualified chemists and biochemists at RASL to be trained across all aspects of drug analysis. But people and culture is very important to maintaining the fight against doping and, of course, for that, our accreditation. Just to summarise, in our laboratory model, of course, we have racing at the core, but RASL has always had a mandate for research and development, and that in the first 25 years of the company was in small molecules. Human urine, as I mentioned before, and oral fluid for external revenue work. From the small molecule R&D, as I mentioned, we've become accredited in investigative testing. More recently, the last 12 years, we then established a biological research unit, bringing in expertise from medical research to tackle large molecules. And then the work in investigative testing is, as I say, enabled us to move into supplements and into human hair analysis. What you can note, though, is those two areas of research and development which aren't currently accredited, and that's where we see a future development with accreditation of R&D activities in accordance with the OECD and GLP principles that NATA offer. This can cover everything for our in vivo pharmacokinetic studies through to biomarker monitoring and ad hoc drug detection studies. And also we expect in the coming years an Australian standard for hair testing is likely. Just to finish, I'd like to acknowledge Dr. John Vine, our founding director of RASL, an absolute maverick of organic chemistry, mass spectrometry and accreditation. He was our founding director, then also scientific consultant for the last 10 years of his life, where he'd had his own bench in the R&D lab, his own Orbitrap Explorus 120, come and play with his chemistry set 2 days a week, Tuesdays and Thursdays. Importantly, he was a NATA technical assessor for 35 years. Many of you would have come across John. Conducted more than 100 assessments in sports anti-doping and workplace drug testing. So, Vale, John.